Good morning, everyone. I'm Chelsea Platsky, Head of People and Strategy for the Global Institute for Food Security, or GIFS, at the University of Saskatchewan, and I will be your MC for today's program. On behalf of everyone on our team, welcome to today's seminar. We're excited to connect with you from our base in Saskatoon, Saskatchewan, Canada. As we gather here today, we acknowledge we are on Treaty 6 territory and the homeland of the Métis. We pay our respect to the First Nation and Métis ancestors of this place and reaffirm our relationship with one another. It's great to see so many people in the room and online, and happy Global Biotech Week. For those who don't know, we are currently in the middle of Global Biotech Week, an international effort to raise awareness about the positive impact of biotechnology and celebrate great science. And we have a lot to celebrate here in Saskatchewan. The province of Saskatchewan is home to more than 30% of Canada's biotechnology activity, encompassing world-class universities, research parks, institutions, established businesses, startups, and of course, a talented community of researchers, leaders, and innovators, many of which are in the room and online today. This week is full of events celebrating our community, and we're excited that this event can be part of it. So thank you to everyone here for your contributions to advancing biotech innovation, productivity, food security, and more. There are lots of other great Biotech Week events that you can join, including Biotech at the Market, a networking and trivia event on Wednesday evening here in Saskatoon. You can learn more about that and other events at Global Biotech Week, sask.ca. Thank you to those filing in. There's a few more seats at the front, so please help yourself. Today's seminar is a perfect fit for Global Biotech Week. Over the next 2 hours, we're going to hear from great scientists and explore exciting new technologies and capabilities that we've established here in Saskatoon. Our topic, engineering, biology, and automation, is something we're extremely excited about at GIFS, where we have built Canada's only biofoundry dedicated to advancing agriculture and food innovation. In our one-of-a-kind facility, biology, automation, and machine learning are combined to unlock a new scale of research and product development. Today, we have expertise and advanced automation workflows to execute thousands of parallel experiments with precision and speed. Just think what's possible. To quote our platform lead, Dr. Ben Scott, who you'll meet in a few minutes, get used to thinking at scales of thousands, millions, and billions of evolving systems. At GIFS, we can design and construct new microbes, proteins, enzymes, and small molecules to help you solve complex problems, enhance products, and create valuable new solutions that make an impact. Think of highly targeted fungicides and herbicides, probiotics for soil and animal health, enzymes to unlock value from byproducts and other innovations that enhance resiliency, nutrition, nutrient efficiency, and more. And that's a small sample of what's possible. There's so much potential, and we're looking forward to learning more about your project, business, or idea. Of course, we didn't get here overnight. This has been a multi-year journey that's involved extensive engagement with stakeholders, significant renovations to our facility, and the tremendous effort of our team, many of which are in the room today. Today, we're excited to share that our renovations are complete, and we are now providing technical services to researchers and organizations. If you're new to GIFS, we hope you're as energized about innovation and collaboration as we are. Collaboration is at the foundation of everything we do at GIFS. We are a unique institute at the University of Saskatchewan with a mission to work with partners to discover, develop, and deliver innovative solutions for the production of globally sustainable food. At GIFS, our ambition is to be your preferred partner, bringing established expertise and leading edge technologies to help you accelerate your work. Today, we are collaborating with researchers and organizations through diverse activities, including our suite of world-class technical services that span engineering biology, genomics, transcriptomics, bioinformatics, and more. Our team has become renowned for high-quality genomics, data generation, and analysis. And earlier this year, our sequencing and genotyping laboratory was named the first Revio PacBio certified service provider in Canada. We are building on the success as we foster new connections with our engineering biology services, through which we will support strain engineering, cell growth and phenotyping, proteomics, and metabolomics. Finally, I want to give a special thank you to many organizations and institutions that invested in our engineering biology capabilities, including the Canadian Foundation for Innovation, Prairies Economic Development Canada, Agriculture Development Fund, and Innovation Saskatchewan. I know representatives of many of these organizations are with us today, so thank you. Thank you for supporting and sharing our vision and working with us to advance agriculture and food innovation. I also want to acknowledge Agilent, our sponsor for today's event. Agilent has been a great partner to GIFS as we established our engineering biology facility, and we're grateful for their support today. And thank you. We have many of them in the room today with us and some online as well. You will acknowledge we have a table where we will be able to learn more as well. Later in today's program, as I mentioned, we'll learn more about some of their technologies. And of course, those joining us for a tour this afternoon will get to see it up close and personal. Before we begin, I have a few short housekeeping items to review. Our program will wrap up before noon here in Saskatoon, so in just under 2 hours. We will proceed through the program without a break. Water and coffee are available at the side of the room, and washrooms are available just outside this room and to the right down the hall. For those of us in person, we hope you can join us after for a complimentary lunch following the program. Those joining us for a tour after the program can meet us at the registration desk at your allotted time. A representative of GIFS will take you to our facility, which is just a short walk away. If you didn't sign up for a tour, please check in at the registration desk and we'll try to confirm a time for you. We have 2 presentations planned and we will be doing a short Q&A after each session following the talk. So if you have any questions at that time, please raise your hand and we'll bring the microphone to you for those in the room. And we'll get to as many questions as we can within the time allotted. For those online, we'll facilitate all questions using the Q&A function. Please enter your question into the Q&A and a moderator will read your question for those in the room. If we can't get to your questions today, please reach out to us at info@gatebygifs.ca and a member of our team will follow up with you. We also want to connect with you online. We'll be sharing highlights from today's session through LinkedIn. If you're active on LinkedIn, we encourage you to do the same and tag the Global Institute for Food Security. It'd be great to connect with you and see what your key learnings were from today's conversations. At this time, I'd like to introduce to you Dr. Ben Scott, GIFS Engineering Biology and Automation Platform Lead, to discuss how GIFS uses automation to engineer biology at scale and share insights from a protein design project at GIFS. Ben joined GIFS in 2023 to lead the engineering biology platform. He holds a BSc in life sciences, an MSc in medical sciences, both from McMaster University, as well as a PhD in cell and systems biology from the University of Toronto. His publications cover topics including synthetic biology, protein engineering, laboratory automation, bioinformatics, and science policy. Prior to joining GIFS, he completed a postdoc at the National Institute of Standards and Technology, or NIST, and served in business development at the Concordia University Genome Foundry. Ben has been absolutely instrumental in leading the development of our engineering biology platform, and it's truly been remarkable to watch his team and the plan come together. Please join me in welcoming Ben.
Thanks, Chelsea. It's a really kind intro. Before I start, I just wanted to acknowledge another reason why I'm here in the room today. And that reason rather, is our CEO, Stephen Webb. Steve recently announced his retirement after over 30 years in the ag and food space. That included academic and then industry experience at Dow and then Corteva. And he started here at GIFS in 2019. And for those of you who knew GIFS prior to Steve and those who know it now, I think you'd all agree that it's a completely different institute. He brought in a vision for engineering biology that just didn't exist. He, over his career, kind of saw the future coming and recognized the importance and the impact that it could have in food and agriculture. And he's really the reason why I'm here and the reason that the institute created the engineering biology platform. So I just wanted to acknowledge, yeah, his support there. Okay. So as Chelsea talked about, I'm going to be talking about some of the automation that we now have in the lab and the capabilities, but then framing that around a very specific project. And actually, what I hope you get to take away are some of the lessons that we learned along the way. So it's not just an info dump or a data dump about all of the cool graphs and all of the cool robots and equipment that we have, but yet you'll actually get to learn something and impart that on your own research program. So my favorite definition for engineering biology is using biology to build useful products. And I like this the most because it's something that I can explain at Thanksgiving to my family when they have to ask me again what it is I do. And that's the definition that I always go back to. What that actually looks like is actually a pretty significant potential for the world. So this is the estimated global market size in the next 15 years. And importantly, what you'll recognize, hopefully, is that ag and food represents about a third of that market. And although human health deservedly gets a lot of attention, ag and food is kind of the unsung hero here, and the potential impact that we could have here is enormous. When I try to think about this in real terms, it really comes down to what is the end product? What are people going to use in their day-to-day lives? And that can look like a plant, it can look like a microbe, it can look like a protein, enzyme, small molecule, with a whole bunch of different applications that each one of these can accomplish. Some of these I'm going to be talking about today. Some of them you might already be working on, and if you are, I'd love to work with you. And some of these are more aspirational. But I really wanted to kind of drive home just all the different ways that engineering biology can impact food security. Something that's not often overlooked, but something that we take very seriously as well in our lab is that improved reagents. So what we're actually feeding into this pipeline also contributes to engineering biology for food security. So that might be a new nuclease, it might be a new way to synthesize or assemble DNA, it might be a new way to produce a protein or extract a protein. It might even be a new microbe that no one else has used before as a new chassis for producing your protein or metabolite of choice. And so, as Chelsea mentioned, we're the only engineering biology platform dedicated to a new scale of food and agriculture product development. The way that when I first started at GIFS, I basically made this slide and figured, okay, I'll find equipment that can do this and then find the people and so on. But this is the way that I tried to frame things, which is that we needed equipment and people that could engineer strains, conduct cell growth and phenotyping, as well as metabolomics and proteomics. And fundamentally, at least in our lab, we don't think of each one of these as separate. We have a small team, which means that we all meet together and talk together. And it also means that each one of these suites of equipment needs to be interoperable. And although they're like, hexagons here and they might look kind of separate, we really think of the equipment and the people here as one team. And getting to the point of the talk, which is harnessing automation, again, I could show lots of cool pictures of robots and really tiny droplets and things like that, which is really interesting. But what it really means fundamentally is cycling this design, build, and test cycle and asking bigger questions. So being able to build more and test more with automation, being able to, sorry, it's a little slow uh, cycle through this faster. So getting to your desired results faster. And then something that I would challenge all of you to think about is asking bolder questions. So specifically being able to ask and answer questions that you could not do so using just manual methods. And I know that sounds maybe a little esoteric, so I'm going to show you data and demonstrate exactly what that looks like. Um, and so, yeah, again, lots of cool, pretty pictures of robots, lots of cool equipment. You're going to see some of that equipment, hopefully, if you come on a tour, so we can show you what that means. But I almost deleted this slide because I don't know, it looks like kind of marketing material. I'm not a marketing material guy. I'm a scientist. But I left it up because I realized there's a really important piece here, the design and learn. So what you'll see in the lab is mostly focused on the build and test, and that's mostly what I'm going to talk about today. But an equivalent half are the design and learn. And something that's really unique at GIFS is that we have these these technology platforms. Engineering biology is one of them, but we also have data management and analytics as its own separate team with dedicated experts. And that's really where their expertise lies in the design and learn aspects. And we need both halves, and that's two different teams. And for me to be able to rely on that team and have them by, an instant message away is a incredible opportunity for me personally as a scientist, but as an institute, I've never worked at somewhere else that has something like that. So left that up in acknowledgement, and I will show some of their work as well because it's pretty cool. So that was the really cool marketing picture. You're not going to see this anywhere else, and you won't see it today because all of this is done. But over the past year, we completely gutted the space. We installed new electrical, humidity, which in Saskatoon is extremely important, and new ventilation as well to support all of this equipment. My favorite picture, it looks really understated, but it's the top one in the right, where you can see all those different cutouts in the wall. So those are just electrical outlets. But I spend 75% of the time in Toronto. So what I had to do is exactly measure down to the millimeter where those electrical outlets were going to be put in the wall so that we could ensure that we could actually plug in our equipment. And full credit to our contractors who executed on that. But I went back and measured all of that, and it was down to the millimeter where they needed to go. So anyway, that picture always makes me smile. And then the equipment that actually went in that space is shown partly assembled in the bottom left. All right. So this is more marketing photos, but these are real photos that showing the team, the equipment, the kind of the high quality caliber equipment and people that we have here. So what can we do with this? I'm going to frame this mostly, I think the next slide. Here we go. I'm going to frame it around a project. This project we actually started approximately the same time that we broke ground on building out the institute. So it's been an interesting past 18 months where we've been taking receipt of equipment, installing it, getting trained on it, and then also trying to generate data at the same time. And I wouldn't say it was quite a sprint. We've been very deliberate about it and planning out every step, so it happened very smoothly. And each time we got a new piece of equipment installed, we started generating data in service of this project. So this project was supported by the Homeworld Collective. They're a nonprofit based in the US to support climate-focused research. And we convinced them that our project was the right one because it was one that was focused on agriculture, and it was one focused on a pretty incredible scale of a challenge that we're trying to address. So that scale is illustrated here. I love this photo because I'm a geography nerd. The top photo here is actually, you can see this on your cell phone if you're so curious. You can go to Google Earth and look up what this is in the middle of the Sahara Desert. This is a 100-kilometer long conveyor belt that you can see from space that is simply constructed for transporting phosphorus-containing rock from the middle of the Sahara Desert to the coast in Morocco. And phosphorus is such an important part of Morocco's economy, it constitutes a significant part of their GDP. And that phosphorus containing rock is then transported around the world. And we can also see one of the end products of that from space, which is an algae bloom in Lake Erie, not far from where I live and grew up. And that's an unfortunate kind of byproduct of modern agriculture, is that this fertilizer tends to run off and other organisms benefit from harnessing that nutrients. So I really like this because it ties sort of two parts of the world together that you maybe wouldn't have thought of. And it also demonstrates the scale of the challenge and potentially the scale of the solution that we're hoping to address with our project. So phosphorus fertilizer. So there's no way around it. All agriculture needs it. You can't just not put it on a field and expect to get good quality crops. But as a result of its production, it generates greenhouse gases. It also, you can imagine, generates greenhouse gases being transported from Morocco around the world. And then it creates these algae blooms. So really not, an ideal product if we were to be able to remove those two problems. It turns out that phosphorus in the soil is actually pretty abundant. So organic phosphorus, which doesn't come from a rock that you have to dig out of the Sahara Desert, but it's typically locked in this molecule called phytate, which is shown on the screen here. And that comprises up to 50% of the organic phosphorus in soil. So there's lots of phosphorus in the soil. So why do we have to rely on fertilizer? The big reason why are all of those negative charges. So if you have any memory of high school chemistry, you might be able to remember that little sign means negative. So that means negative. And then there's other ones that are positive that like to interact with each other. And those positive ions happen to occur in soil as minerals. And what this means in practice is that this really, really highly negatively charged molecule is getting absorbed and binding to all of these positively charged molecules. And that means that biology cannot access it. It essentially forms a mineral complex in soil. Now, this being such a great resource, all life needs phosphorus to be able to grow. Life tends to find a way, and there's a family of enzymes called phytases that can release phosphate from phytate to make it biologically available. So we figured, okay, if we can create a phosphate releasing enzyme, maybe we can turn this into a product that makes this bioavailable to plants and then reducing the need for fertilizer. So that was our goal with this project. Sounds simple, right? So all we needed is an enzyme that works at a neutral pH, is a low temperature, especially, in Canada, it doesn't get super hot here, and also is able to liberate these metal phytate complexes. And despite the best efforts of evolution, there's no natural phytase that checks all of these boxes. And so we figured, okay, we're engineering biologists. AI and protein design looks pretty cool. Maybe we can just design one. And that was the purpose of this project. So the goal here over these 18 months was to develop methods for designing thousands of phytases with desired function. That function looks something like this. So we want to be able to improve its activity, improve its pH and temperature optima, and then improve its stability. If we want to think about applying this in a field, it has to be stable, it has to be active for a long time. And as I alluded to at the beginning, I am going to touch on the design aspect briefly, but the majority of the rest of this talk is going to be about the design, sorry, the build and test. So once we have those designs that come out of a computer, as impressive as that might be, we need to test them at the end of the day and make sure they have the function that we're interested in. All right. So what does it mean to build a protein from scratch? So we're not, omnipotent. We need to start somewhere, right? And we knew that there's natural enzymes that have this function. So we looked at across nature and said, okay, these ones look the most interesting to us. We knew that there's a particular part in that protein that is really important for function, and that's where that molecule is bound, shown in green, around that little orange and purple part in the middle. And then there's also parts in that protein that are important for its stability. And these are disulfide bonds. So these are two amino acid residues that have formed a covalent bond. And we knew exactly where they should be in the protein structure. We then took a whole bunch of these natural sequences, aligned them, and then told the AI algorithm to say, okay, of all of the residues that are here, those green ones are really important, so don't change those. But then learn from what nature has tried to change the rest of those residues shown in gray. And then it spits out this novel phytase, and we kind of hope that based on some, in silico computer-based filters, it's going to have the activity that we want, the pH and temperature optima, and the stability that we want. And we can basically do this forever. There's no limit to this. It's just based on how powerful your computer is, how long you're willing to wait. And so we figured 20,000 sounds like a good number. It took us a little while to figure out this whole pipeline. I'm simplifying it a lot, obviously, but 20,000 seemed like a lot and seemed like a good number for us. Here I'll just acknowledge Aaron's contribution to this project. So Aaron is that beautiful mind that I could message when I'm in Toronto and he's here in Saskatoon and say, hey, protein design, here's the latest paper. Can you figure out how to integrate this with this pipeline? And he messages back, sure. And then I don't hear from him for 2 weeks. And then when I meet him again, he's got it there and then the next step after that. So kudos to Aaron. Really, really fantastic researcher. So he's designed these 20,000. Here's an example of what that looks like when we predicted the structure. So that crystal structure is shown in gray. And then one of these 20,000 that I literally just picked randomly is shown in blue here. And the really important thing, other than, it's kind of mesmerizing, it looks cool, right? But the really important thing are those two numbers up at the top. So the sequence similarity to the natural protein is actually very low, but the structural similarity is very high. And that's really important when you want to think about building something that nature has never tried before. You need the structure to kind of look the same, right? In order to bind that molecule, have that enzymatic function. That's one of the sort of dogmas of protein science, structure equals function. But the sequence that actually gets you to that structure is very, very different and does not exist elsewhere in nature. So that told me, okay, we're on to something certainly different, but is it going to work? That comes to the first lesson that I'll try to impart upon you, which is that your build and test capacity must match what you can do, the design and learn. So you can run that computer overnight, you can run it for 2 months, but at the end of the day, you need to be able to build and test those. And that's really where automation starts to really come in handy. So part of that automation comes down to the physical components that we're actually going to test. And one of those, as you can imagine, in engineering biology is the DNA. So how are we going to get DNA into a cell in high throughput so we can test these thousands of different constructs that a computer has built? There's two different types of cells that we work with, E. coli and yeast. And there's different variables indicated here that we know are important for getting that DNA into a cell. And we could do this manually, but no one wants to be doing this 20,000 times. So we started to put this robots that we had installed at this point to work. Sorry. There we go. So what does that robot look like? It looks like this. It's kind of a blue and black box with a bunch of metal parts. And the advantage with this is that we selected the components of this with this purpose in mind. So it has to be able to control temperature, be able to move very small droplets of liquid around in a sterile environment, and have very precise control of that blue arrow, which a human never thinks about. A human never considers how far away should that droplet be from that dish when I put that droplet on it. Like you never think about that. If you're in the lab, you put it on there, you kind of shake it around, you walk away, and you hope it grows. But a robot can't see that plate. It can't actually know, that there's even a plate there until you tell it that there is. So how does it know how far it needs to go to put that droplet on? And this is acknowledgement of Josh. So Josh is our automation engineer. He thought of all of these variables and then said, okay, I can program it, but what about if someone else wants to use this and someone else who's not an automation engineer? So he built this system that's shown on here, which you can just input the different components of these variables to then get to your desired output, which looks something like this. So after quite a bit of trial and error, we were able to show confidently that we can put these DNA molecules into both E. coli and yeast. And to put this into perspective, this is a Petri dish about this big, and which is not much bigger than a single Petri dish. And that would mean instead of doing this 96 times, where a stack of that would probably be about this tall, and someone would probably, quit at the end of the day if they had to deal with that, understandably. This can be done. You load the deck with media, cells, DNA, and you can walk away. And it's done when you come back a few hours later. So that now we can iterate this on a few hundred per day. Okay, so that's getting the DNA into the cells. What happens after they're in those cells? So we've got these thousands of colonies. We've got all these constructs. What are we actually going to do with them? Well, okay, they grew, but now we need to move them to media and then maybe extract their DNA or protein that they're producing. So Matthew, shown here, has kind of become our resident expert and our colony picker, which is specifically designed for doing that. So picking colonies in a sterile environment as quickly as possible and with the precision and accuracy of automation. So you're not, picking a colony and then trying to remember, was that the biggest one? Was that the most green one? Which well did I put it into? All of that is now automated. And again, you load it with your plate and media and you can walk away. All right, so I'm actually going to take a step back. So that was assuming we had the DNA and we were ready to put it into the cells. But at the same time that we were developing this transformation procedure, we wanted to make our assembly procedure as high throughput as possible as well. We knew what it needed to look like. And if we could just synthesize that efficiently and cheaply 20,000 times, we would. But until gene synthesis is, a penny per gene or a penny per construct, we can't do that. So that means some level of assembly is required. The first stage was 8 pieces, so these 8 different boxes. You can think of them as 8 different Lego bricks. And then there was another piece that we had to put in, which was actually our phytase. So once we had those 8 pieces, then we needed to put one more in. So all in theory is fine, but to be able to get to the 20,000 scale, not easy to do. And so we have a piece of equipment. This is a cartoon, and I like this mostly just because of the blue illustration here. So rather than using tips, plastic tips, to move liquid droplets around, it actually uses sound waves to bounce or flick a droplet from one plate to the next. And it sort of shouldn't make sense. It shouldn't work. But just on the properties of surface tension, even though that plate is upside down above it, and those small droplets are so small, it sticks to that plastic, and you can miniaturize these reactions down to a few nanoliters if you so choose. And Tongbo led this work. She was the one where I could say, okay, Tongbo, this is what we need. Figure out how this equipment works. Let's see what happens. And she pushed it even further. So this is an assembly efficiency of 2, 4, 6, and then up to 10 pieces for the DNA assembly. And you can definitely see the efficiency starts to drop off, as you might expect. But that red line was kind of our bare minimum. And even at 10 pieces, we're able to assemble these reactions. So this is being able to miniaturize our reactions 20 to 40 fold. And this is minutes to prepare 384 assembly reactions. And then to close the loop on that, she then took that automated transformation procedure and showed that we could get a ton of colonies, even out of a half microliter reaction. So that brings me to the next lesson here, which is that workflows require validated modules. It's not as simple as just pushing a button, even though I'm making it sound easy. You need each one of those parts, the build, the transformation, the colony picking. Each one of those we validated on its own before we're able to stitch them together and thought about what is actually capable, what are the pain points, and you really can't just theorize about it. You have to do it. So this is the result of that work. Okay, so what about those phytases, right? Well, hey, turns out we could measure function in some natural versions of them, which was pretty exciting. So we're taking a gene from some other organism, putting it into yeast, secreting it, and then measuring how much phosphate is released. And we could see some activity. Those big blue bars means that there's some phosphate being released, and that matched more or less what was in the literature. So we thought, okay, this is cool. But this took 4 hours to get 96 samples, which is, pretty good, but also a bunch of harsh acids. So it's actually not a very safe assay, and we couldn't use any of our liquid handlers for this. So this would mean that one of our team members would be in a fume hood till, the next ice age to just try to pipette out all of these acids and things. So not much fun. So instead, Josh and Matthew developed a fully automated, well, 90% automated, not fully, but 90% automated, 12 times faster assay that does not require any harsh acids using our equipment. I'm just noticing there, that's our first piece of Agilent equipment that's up on the screen. That's one of our plate readers, and become a real workhorse in our lab. And then that gave us not only an ability to measure in high throughput, but also to start changing some of the variables that we were actually interested in. So starting to change those pH and temperature optimas, which we really couldn't do using that classical assay. So this revealed something really interesting, which is those two versions down on the far right there looked like they had activity at both 40 and 20 degrees. So something that no one has really reported before, and something that we could use in our protein design pipeline to build those proteins with desired function. This, by the way, N of 12, N of 9, that was all performed in a matter of weeks once we had that assay developed. So we could iterate on that very quickly. So this brings me to the next thing I'd like to impart upon you, which is that I think this is probably my favorite lesson, I would say, which is that I really want everyone to constantly challenge scale. So ask yourself, why am I running this number of samples? Why am I running at this volume? Why am I taking these time points? Because I would guarantee you that automation can get you to a different scale for one of those, at least. And 192 samples sounds like a lot, right? Like that's more than 96, but it's not 1,000. So I decided, okay, we're going to keep challenging that scale. And we knew that all life needs phosphorus to grow. So if we could tie the function of that enzyme to growth, we would be able to simply plate out cells or grow them in media. And rather than some sort of complicated enzymatic assay, we'd just look for which ones grew. And that is the most high throughput, cheapest way to measure biological function is, does it grow or does it not? And again, I challenged Tong Bo with this. We had some ideas based on the literature, but no one had really shown this before. This was some of the first data that we got out of this, which was a very happy day in the lab, which was to show that the ones that are producing phytases are able to grow, and the ones that we specifically mutated cannot. And with the help of these plate readers, we can scale this very, very easily, where you're just picking colonies and growing them, and you can walk away. And so we can iterate at about a scale of 576 a day, getting these really quantitative growth curves. Those growth curves, I had to delete the slide. I had to cut sometime somewhere, but it pretty well perfectly matches the enzymatic assay. So this is getting to, again, a completely different scale. And using a combination of our automation, so going from our colony picker to our high scale or our high capacity incubator, we can feed this incubator plates. And by just taking an endpoint measurement, which is to say, we don't care about the full growth profile, let's just look at the beginning and the end of growth, we can scale this up to 6,000 phytases screened in a day. All right. So I introduced this to you at the beginning. I've introduced different parts of these design, build, test, and learn. But what if we could combine some of these? So I've shown you individual modules. I've shown you a lot of interesting equipment, but each one of those still requires someone manually moving a plate or coming in between the hours, 8:30, 4:30, or 9 to 5 to do that assay. Could we automate this so that you could really close that loop and design something and then walk away, and then you've got something to learn from after you've walked away? So that's the holy grail of automation, ultimately. I won't say this is completely validated, but this has been installed in the lab, and this was purpose built so that we could iterate on this scale and close that loop. So what does this green box do? This is probably the most technical slide, and I'll just pause here for a moment. It is a liquid handler, fundamentally, with a bunch of different pieces of equipment from about 4 different vendors that have been installed along the back. And by virtue of its automated robot arm, you can move these plates across each of these incubators, plate readers, sealers, peelers, such that once you've loaded the deck with media, microbes, and plastics, you are able to walk away and get that data, a day or two later without any other intervention. So that data can look like a growth curve, it can look like fluorescence, it can look like a color change, or just looking to see which ones grew and which ones didn't. So when we want to iterate on that, oh, and most importantly, it can select which of those after you've run that experiment and then passage them into the next experiment. So you can iterate on this in a closed loop system. So I couldn't find like a more descriptive photo. I could probably talk about just this piece of equipment for the next hour. So I hope that you have a chance to see it on the tour. All right, so let's say we found that one phytase. What do we do with it? Well, I've only talked about the milliliter or nanoliter to milliliter scale. And obviously at that scale, which is maybe about the size of a raindrop, we can't apply that to a field or even a greenhouse. So we've also installed these new bioreactors that can go up to 10 liters. They offer OD, pH, and off-gas analysis so that we can really scale from that nanoliter all the way to a liter scale in the same institute. Okay, so I've talked a little bit about this molecule, but I kind of didn't tell the full story. So these enzymes are pretty good, but they really struggle with releasing phosphate from the last 3 positions. So there's IP6, IP5, 4, 3, 2, 1, and then none. If you're only releasing 2 out of those phosphates, or 2 or 3 of those phosphates, you're leaving about half of that phosphorus on the table. So we wanted to understand, is there a phytase out there that can release all of that and increase the amount of free phosphorus 2x? And like I said, natural enzymes really struggle with this question, but we looked at this as another opportunity for protein design. But how are we going to measure that? So not every question, unfortunately, can be answered by growth, fluorescence, or color change. And that's really where we start to talk about metabolomics and proteomics. And those are able to now answer questions about how much product is there, what side products are being produced, of those hundreds, maybe thousands of strains, which one is producing the most, and then amongst those proteins, which ones are present and which ones have post-translational modifications. And you can get clever maybe in using growth, fluorescence, or color change to answer those, but fundamentally you need a different type of technology, and that's where metabolomics and proteomics comes in. So with this suite, we aim for quantification analyses of these molecules at a scale of thousands of samples per day. This is some examples I already kind of talked about. And the equipment, which is all Agilent, comes in the form of the rapid fire automated SPE. So rather than a traditional LC system, this uses a cartridge that the samples are essentially shot through at high pressure to offer some type of crude separation in that and sample prep. So it's not going to give you, the cleanest data, but at the scale and speed that we need to iterate, there's no other technology that can kind of achieve that scale. So this is up to sort of 8, maybe 16 seconds per sample versus the minutes that it takes per sample of a traditional HPLC, which we also have. And both of these injection systems can be connected to our high sensitivity MS, which is a triple quad. So when you want to answer down to, quantitate which molecule you're interested in is actually in your sample, as well as a QTOF, which looks at a more untargeted screening. So what other molecules are there? What are the byproducts, off products of that metabolic pathway? So by having this range of capacity and range of scale, this is certainly, again, in my career and the institutes that I'm familiar with, this does not exist anywhere else, coupled with all of that other engineering biology technology. So what have we used this for? So this molecule does not play ball with LCMS. So it's highly polar, it's negatively charged, so it makes ionization pretty difficult. And in addition to that, we're interested in asking a question about the difference of a phosphate group being there or not. And that's not easy to do with this piece of technology. I'm looking at Agilent because I talked to you guys about this and you say, oh, that's good luck. I appreciated that, the honesty. And so I said, George, you've just started here. You started here in June. Um, this is the challenge that I'm giving you. Welcome to GIFS. And he said, sure. So he took the equipment that we had, and over the last less than 2 months, he's developed a method for separating out each of these species that we're interested in. And I'm incredibly impressed, and I'm incredibly also excited for the sort of questions that we can ask now and answer, which is which of these enzymes can actually push those limits, those natural limits that evolution has not explored. And that brings me to the last, and I think the most important lesson, which is that high quality equipment needs high quality people. So we cannot plug in this equipment and push a button, and I can fly back to Toronto, and then it spits out science. Like that's just never going to happen. So having the team sitting over at the table over here, having the support of the institute it's world class, and that really fundamentally comes down to the people. With that, I'd like to thank you for your attention. These are all the fine people here, as well as the support from these great funders. Thank you.
Thank you, Ben. And I have to say, there truly is world-class expertise. And it's important to note that you, online and in the room, have an opportunity to tap into this. So we are offering an award, and many of you would have seen this as part of your registration that GIFS is supporting an award opportunity for those attending today's seminar. The winner of this award will receive a discount up to $5,000 on strain engineering and DNA assembly services at GIFS. We'll share the application link again at the end of this program. A huge thank you, Ben. I have the pleasure of working with all of the people that you saw on screen, and I am constantly amazed as I continue to hear about the leaps and bounds that they are able to make on a day-to-day within the Global Institute for Food Security. So huge thank you. As promised, I want to open the floor to those in the room and online. We do have a few minutes for some questions. For those in the room, if you do have a question, please raise your hand. And I know we have a colleague here at GIFS that will bring the microphone to you. Those online, please feel free to type your questions into the Q&A box.
I have a question. So, what platform or website did you use to generate the 3D protein images?
Oh, that's PyMol.
Okay.
Yep. Good old PyMol, right? Yeah. Yeah.
Great presentation, Ben. I'm wondering, so you're building these proteins, you're expecting them to be structurally sound, and there is activity in a controlled environment. Is there a plan once you've decided your delivery system to test that effectiveness? And have you tested that yet?
I have not, but we've got great greenhouses at GIFS, which you probably won't get to see on the tour, unfortunately. But the plan is to be able to produce those phytases at the 10-liter scale, purify them, and then test them in the greenhouse. So having all of that kind of in-house is, again, dream come true as a scientist.
Is the application of that in the greenhouses like spraying the enzyme through, and is it going to permeate through the soil? Is it just going to tackle the organic phosphate at the top of the soil? How do you know it's going to properly get as much?
So that's why no one's given us more money because I haven't been able to answer that. But material scientists, if you know a material scientist, I want to talk to them. If you know someone working on agriculture residues that can already be added as a biofertilizer, I want to talk to you because we have some ideas about how to functionalize those materials and residues so that we could get them deeper into the soil and we're not just sort of spraying and praying with the enzyme, literally. Yeah.
The last one, probably. I just have a question. Like you talk about the enzymes, yes, which kind of exists in nature, right? Do you think is it possible to do the same thing just with the enzyme kind of which not exist in nature, hypothetically. For example, like I think Baker Lab, like famous Baker Lab, they are doing something like this. And is it possible actually to do in this, like in this environment?
I mean, that's what we're hoping. Like the reality is sequence space, protein space is essentially infinite. And I don't believe that nature has solved every problem. So that's the idea behind protein design. And in those 18 months, hopefully I made it look impressive, but within those 18 months, it was fundamentally to establish that protein design pipeline in-house so we could apply it to other families of proteins as well. So if you have a protein that you're interested in, particularly enzymes, binders, things like that, I'd be really interested to talk to you because the fundamental technology is essentially the same.
There is a question online. Open tools are critical for developing the bioeconomy across Canada. How is GIFS working to support open tools?
Open tools. I love open tools. So we need tools that we have freedom to operate with. And that means they're not as restricted by traditional MTAs. And so I'm a big proponent of developing and using tools that people can use for those commercial applications. So we benefited from receiving some of those tools. We want to be able to return that favor and share some of those tools as well. And that fundamentally comes down to, in my belief, those DNA parts that we're using. There's no patents associated with most of those. There's no restrictions other than a traditional MTA. And I believe that, if we all kind of contribute towards this, we can all succeed. So yeah, big proponent of open.
Perfect. Well, thank you so much. If anyone does think of a question, you're more than welcome to ask it when you are on the tour. If you are signed up for a tour as well, please don't hesitate to reach out to us through our website, info@gatebygifts.ca, if you do have any more questions. And again, a huge thank you to Ben. Absolutely great presentation. So thank you very much, Ben. Perfect. Well, at this time, I have the pleasure of introducing you to our next speaker, and I'd like to introduce you to Dr. Karen Yanell, who is joining us virtually. Karen has a PhD in analytical chemistry from Purdue University. She is an expert in mass spectrometry and small molecule and biological sample analysis. Karen is an LCMS Omics workflow specialist with Agilent. Our team has worked closely with Agilent over the last few years as their technology is helping to power many of the technical services at our engineering biology team is providing, which you saw in Ben's slides. And for those of you joining for the tour, we'll get to see it in person. Lots of Agilent technology. So today GIFS is equipped with, as I said, multiple leading edge instruments from Agilent, including advanced triple quad and rapid fire mass spectrometry systems. Today, Karen will discuss end-to-end workflows for such systems, exploring sample preparation strategies, data collection, and analysis. Please join me in welcoming Karen.
All right, can you guys hear me okay?
Yes, we can.
Thanks so much. All right, very happy to be here virtually, although kind of sad I can't visit the tour because it sounds like your guys' lab is very nice. And congrats on being done with the construction. I'm here to explain some of the LCMS technology that's in the space and how to use it for metabolomics, lipidomics, proteomics workflows. You have access to a triple quad. It's a great triple quad, the 6475, and an LC-QTOF. So I'm actually just going to begin my talk really going in, and this is me trying to bring everyone along in this talk to understand what these instruments do and what they can do to get you data that's going to help your projects. So I like to start my talks off though with kind of some background, really broad background on pathway interrogation using these two tools. I get a lot of people coming to me saying, Karen, I want to do untargeted discovery. That's where you're going to collect data, metabolites, lipids, peptides on all of your analytes. And this is where collecting that data with modern high-res tools, and this is our Revident. Let me go into Pointer. This is our Revident LC QTOF, which is in the lab you'll see today. This is where collecting data on a modern QTOF is pretty straightforward. It shouldn't take extreme expertise to collect good data. What does take some consideration, though, is now that you have all that data, how do you extract the features, do the statistics, do the identification, especially metabolomics and lipidomics? Identification is tricky. And that's where I will talk about a piece of software called Explorer that makes that easier. But very commonly, when you actually go look at some of the best core labs doing this research, they're not actually trying to discover new metabolites. They understand pretty well what the pathways are. So they collect all the data, but they just go in and they mine the data for the metabolites that they expect to see or that they want to interrogate. So really, it turns into kind of a pathway investigation and not necessarily what is every signal, because a lot of the signals in LCMS kind of are just background noise, to be honest. They're not all biologically relevant. There's a lot of papers discussing that. So this is a very common approach that simplifies the data analysis component. You do need to have some libraries up front, which Agilent does provide. So I will show you that. And we also have a tool called the Screener tool, makes that analysis faster. Now let's go to the triple quad side. I'm going to be showing today the 6495, but in the lab, you guys have a 6475. It's a great instrument, really robust. Everything I'm going to show today applies to that system. When people collect data on an LC triple quad, they expect it to be, it's all targeted data. You have to program the instrument to go and look at the molecules of interest. Many people use this for 5 to 50 analytes, going in and just targeting a couple things. A lot of people do quant with this because it's a very sensitive instrument. It has really nice dynamic range, great for calibration curves and quantitation. However, modern triple quads, like the one you guys have, actually are so fast that you can put hundreds of analytes in one single injection, allowing you to literally look at the same pathway coverage that you are getting from a high-res system. So when we actually do this with customers who have both, they can measure the same pathways in their untargeted approach as well as a targeted approach, as long as they have those databases up front, which is a big component of what Agilent brings to the table. So I am the workflow specialist. My background is in mass spectrometry. I am a method developer to my core. So feel free to reach out to me directly if you do have questions, although I know Ben is there as well to answer your project questions. But when you think about mass spec, you really need to think about every step that your raw sample is going to get, because if you don't put a good sample into your instrument, you're not going to get good data out. So my talk today is basically going to outline some sample prep techniques, some separation methods. These are methods that go along with our supported databases on our mass specs, and the databases are so critical because that's how you're going to get a leg up on the identification workflow. Software, we have a little bit of flexibility to use some software that Agilent provides or some third-party software that's free. And basically just understand every time you are going to do an experiment here, you want to pair it with the right piece of software to get your answer. Underlying all of this, if you guys ever do want your own mass spec, understand that we are an excellent company at providing services. So where I'm an expert in our headquartered lab, we have experts just like me going out to customer labs, training them on all these methods, really trying to get more biologists running our equipment so that they can get answers on their own. So what are our methods? We have metabolites, lipids, and some peptide workflows that can go onto these instruments. And that it is part of the consideration of sample prep, how to do the separation, and how to basically program the instrument into collecting that data. For metabolites, metabolomics is probably the trickiest because of the chemical diversity that you see in the world of metabolomics. Just having so many different chemical properties, having one analytical column and one set of source conditions that are going to measure everything is pretty much impossible. Hillic is a type of chromatography. It's notoriously kind of tricky to do, but we have a very robust Hillic method, very reproducible on retention times. And because of that retention time reproducibility, that's how we can back up our Hillic method, which is meant to be as universal as we can do. We have a method that's very reproducible with retention times, so I can annotate over 500 metabolites. These are polar metabolites across a lot of different matrices that really every biologist I know has been using this method regardless of the matrix that they have. And not every metabolite, though, is going to be seen in your sample. So that's why we say it's a database that you can screen for analytes in your method. That's on our triple and analytes in your sample. That is on our triple and our QTOF, as is our lipidomics method. So lipids, a little bit more straightforward in terms of separation. It just uses a C18 method. Our current method is 763 lipids. We're going to be releasing a new one shortly. Impactfully, this is 44 lipid classes. That's where you get a lot of your biology information, as well as the curation of this method has double bond annotation. That is super important because if you follow our chromatography, you have way more information because we have a collaborator who did all the double bond legwork annotation for you already. That means that you get a lot more information from than you would just from a typical LC-MS/MS methodology. And then on the proteomics side, we basically have two options. On the high-res side, that's where our systems are really great for intact peptide mapping, and we have a piece of software called BioConfirm. On the triple quad side, you can use the same exact triple quad for all three of these, and you can be doing using kits in order to do the peptides. Peptides are a little bit trickier because they're so specific to the biology that you're looking at, which ones you're going to measure. Kits can get expensive, so that's why we also have kind of a discovery workflow I'll mention at the end of the talk today using free Skyline software. And this is an approach to kind of do some peptide discovery on a triple quad that is a very unique approach to discovering peptides. I did want to start, though, like I said, from the beginning, how to treat your samples. Typical sample prep strategy. If someone were to bring me just a raw sample, I don't care what source it came from, I would look at doing a dilution. A protein crash is very, very typical. It's not my favorite, especially if I'm doing metabolomics, because it doesn't remove lipids. Liquid extractions, especially something like Bly-Dyer, that's going to remove lipids for me. Or actually, I can take the lipid layer and run my lipid analysis, and take the metabolite layer and run my metabolite analysis. It's not really automatable, though, because it's biphasic. It's really hard to get automation involved with that. So that's when when they start thinking automation, they start looking at solid phase extraction. It's basically the best sample prep you can do. So I'm showing you it in this list because I'm showing you increasing complexity as you go down this list, but you're also improving your cleanup as you go down this list. If you are bringing an LC-MS person a brand new sample, I would highly recommend you go look at Agilent's sample prep selection guide, as well as these fundamentals. These are two really, really great, this is a really great document that really breaks down key terms all the way to very common strategies. I just don't have enough time to go over everything today. But once you know, okay, I have to use an actual SPE product, the Agilent has a really nice guide in helping you figure out what exact product you should probably do. And the reason you're doing sample prep is that you need to clean up your sample to remove matrix. The more matrix removal you have, the longer your analytical column, which is probably one of the more expensive things on your LC-MS that is a consumable, the longer your analytical column will run, and the more sensitivity you have. So you can always do more sample prep if you're like, oh, I'm so close to seeing my analyte, I just need more sensitivity. Sample prep or analyte concentration steps as part of your sample prep can really help improve sensitivity should you reach the limit of your analytical method on the LC-MS. But I do want to emphasize this because I think it's a skipped over part. Whenever you do sample prep, you have to put that material back into solution. So let's say you do all this sample prep and you dry your sample down, you have to reconstitute it in LC-MS friendly solvents that are going to work with your chromatography of choice. So understand whoever's going to run your sample, if you're doing the sample prep, what chromatography they're running, and you want to make sure that your sample is going to match those solvents, usually the starting conditions. So when I have a customer sending me samples for our Hillick method for metabolomics, I make sure that they understand we need to put it in about a 70% acetonitrile, 30% water methanol solution, because those are the starting conditions. That's how you get really good peak shape of early eluters. I want to show you one example of kind of a sophisticated technique, though, that's combining all three fractions for extraction from the same aliquot. And I'm showing you this with plasma, but we do this with cells, we do this with microbes, we do this with a lot of different biological samples with some adjustments to the protocol. But basically, you take your initial sample and you do a protein crash. You take that supernatant and you put it on our solid phase extraction plate, and this one's called Captiva EMR lipid plate. It comes in cartridges as well. I like plate formats because they're always automatable. We have automation system called the Bravo that can do this, but you can also do this manually initially if you wanted to try it out. This solid phase is really great at extracting or capturing lipids. So the proteins are going to stay on top of the filter, the lipids are going to be captured, and the metabolites are going to flow through. If all you need is metabolomics, there you go. You have your sample that you can dry down and reconstitute in your solvents. But if you wanted to extract the lipids along with metabolites, you can put a second collection plate underneath there, put a second set of solvents that are stronger, and elute off of the solid phase the lipid fraction. So now you have a combo, metabolites and lipids from the same sample. Finally, in 2023, my colleagues who developed these methods for me also realized, okay, we can also extract the proteins. So from the same exact aliquot now, you centrifuge the proteins off over here and then move the supernatant to this solid phase plate, and it does the same thing. Metabolites flow through, put a new collection plate on, lipids flow through, and your proteins are still captured over here that they can be digested for peptide analysis. This is just a novel way of thinking about getting more analyte classes from the same aliquot of sample, which even if you're not limited with sample, can be advantageous for not having biological differences in the aliquots that you're taking from a sample. So from the same aliquot, you can have the same omics extractions for different LCMS analyses. So now getting to the LCMS, Ben mentioned you guys have a rapid fire. That's a really quick way of just doing a quick sample cleanup and injecting something without chromatography. Very good for rapid, it's in the name, rapid fire analysis, and you're just not going to have chromatographic information, which is fine for screening approaches. When you do need the chromatographic information, you have an Infinity 3 bio-LC in or I think it's an Infinity 2 bio-LC in your lab. This is a LC that is amazing for metal-sensitive analytes, which are really, really common in metabolomics, some lipids, some peptides. Anything with the word phosphate, like the analyte that was showing earlier, anything with the word phosphate or carboxylic acid is going to have more issues with chromatography than analytes without. But this is a system that is iron-free, making those analytes have sharper peak shape and more sensitivity because they're not getting absorbed in the stainless steel. Anyone with a stainless steel LC can, that is an Agilent system, can still get good results. You just have to deep clean it more periodically. So this is a great LC for our purposes. And Agilent, for our Omics customers, standardizes this. Why do we do that? Because all of the databases I was mentioning earlier that we like to give out to customers have retention times. And so we know when a customer wants our Omics workflows, how exactly to plumb this LC so they get the same exact retention times as us. So that's where LCs really do become a critical part of the experiment. I mentioned earlier 3 main chromatographic techniques to measure metabolites and then lipids and then peptides. I'm going to go through those really quick here. This is Hillick chromatography. It's not for the faint of heart. I don't recommend anyone try and build their own Hillick method if they aren't an experienced chromatographer. It's not an easy technique to just pick up and be successful with. However, Agilent does have a very robust, like I mentioned, Hillick chromatographic method, which is outlined here. It's basically using a Hillick column with ammonium acetate buffer and acetonitrile buffer. It's high pH, so you get this TCA cycle here. You can measure nucleotide phosphates, carboxylic acids, amino acids, all with this method, all in one injection on that triple quad. And this is a method that you just need to follow the protocols. If you follow the protocols, you'll get the same retention times as Agilent does, which is where our database comes into play. I'd say that this method, because there's no such thing as a perfectly universal method for metabolomics, it's really great for the compound classes here. The only compound class that doesn't do exceptionally well are sugars. If you do want a sugar method, this column can measure sugars. You just have to go to a higher pH in order to get that separation in single peaks. We have sugars in our database. They just are not going to separate as well as other compound classes. So robustness is key here, and let me tell you, we did pressure test this method to basically show across multiple lots of columns, because Hillick actually has manufacturing issues on top of end user issues. So multiple lots of columns were tested with this method, as well as multiple batches of solvents. This is basically to show, look, any user, this was me making the solvent, this was my intern at the time making the solvent, this is a GCMS chemist who doesn't use a pH meter at all in his life. He made the solvents for me. We're basically showing the retention times are going to be all the same for these complex sample, or this a complex sample with all these analytes in it. This is all to prove that when you use our method, you should be getting the same results as us, making this fit for purpose for kind of anyone in an LCMS experiment. Now, you should expect good peak shape from your any chromatographic method. Here, I'm showing a couple different compound classes having good Gaussian peak shape. But with Hillick, again, going back to the compound diversity that you get with metabolomics, some of the peak shapes are going to be a little bit wider than others. So I don't want you to expect the sharpest, most sensitive peak shape for every compound class. It's a trade-off. Hillick is great for separation of polar compounds, and we can make this universal methodology, but GABA here has 0.2 minute peak width, but something like GTP is closer to a minute in peak width. So do expect some variation on the peak widths or peak shapes a little bit, but really this is probably the most expansive single injection method that you can be doing if you're looking to profile metabolites. Lipids get a lot easier. The lipid, but they suffer from double bond issues. So lipids is a much more straightforward C18 method. Again, we have protocols for anyone wondering to measure these on an Agilent LCMS system for you to follow so that you get the exact same chromatography as us. And why that's important is because our database that goes with this, not only is it going to cover all these lipid classes, and we have, this was annotated for plasma, but we have people using this in all different types of matrices. But the double bond annotation was done by our colleagues at the Baker Institute, Peter McHell's group. He ended up basically saying, look, if this is your transition for this PC lipid, you will see all these chromatographic peaks. Those are all peaks with this, that are this lipid chain. The difference is there's different double bond placement of on that lipid chain, and he annotated, his group annotated using all these orthogonal experiments, ultimately using a lot of standards. He annotated all those double bond placements, or at least like 90% of them in the method. What this means is when you use our exact method, you get you not only get to leverage all of these transitions for lipids, but you also have a double bond information, which most LC-MS methods don't give you without you annotating them yourselves. This is just more information for people using our methods. Finally, peptides. Peptides get a lot more straightforward. I'm showing you a 60-minute method here. Triple quad can go really fast. You could probably shorten this. But with peptides, you're measuring a lot more transitions. So every single peptide, you're not measuring one signal, you're usually measuring 4. So this is a peptide kit. It's 375 peptides associated with 375 proteins. Each peptide has 4 transitions and 1 internal standard for this method. So I am looking at over 2,000 signals in this one injection when I run it. what this means is you're basically pushing the limits of the triple quad in terms of how fast it can turn over its different measurements, looking at from one peptide to the next, to the next, to the next. I'll show a little bit more of that when I show you the triple quad data. But basically, this is a much more straightforward chromatogram measuring peptides because they all have very similar chemistry is probably the easiest out of all three chromatographic methods I showed you today. So now that you understand the sample prep and the chromatography, I want to break down our instruments for you, starting with the QTOF. Agilent has a lot of high-res systems, starting very simple with just a TOF. You're not going to get any MS data. You'll just have high-res data. All the way to an ion mobility. Ion mobility actually adds an extra dimension of separation to understand like the shape of a molecule as well as its mass. However, for today, we're going to be talking about the Revident, which is probably one of the best small molecule instruments I've gotten to use in my career. And that comes down to a lot of improvements, not just from the usability of how easy is it to maintain and run, but also the detector and the electronics on this are giving me some of the best data, which is how I'm getting identifications better on this system than I have on previous high-res systems that I've used. So this is for anyone who wants to know the specs, but understand that basically this QTOF, which is a high-res instrument that will give you MS data, and that MS data is how you do structural investigation of what is this signal? Is it a metabolite? Is it a lipid? Is it something not related to biology? What you're really looking for when trying to get these confident IDs is a combination of the resolution, which is, it's going to be 35,000 or a little bit higher depending on the mass. Resolution, the dynamic range, can you measure the small stuff when the large stuff is co-eluting on top of it? Isotopes, so this is the molecular ion plus all the heavy, naturally abundant heavy isotopes that really helps us fingerprint what the chemical formula is, and mass accuracy, so making sure that molecular ion is constantly the same throughout your entire experiment. All these things are done really well in the system. And I want to show you some examples. This was a plasma sample injected over 200 times, was a 7-day experiment. And my colleague, who was new to our lab, she just finished her graduate work. She came in as a postdoc. She used a triple quad, but never used a QTOF before. I taught her the method. I had to go on a business trip. I gave her the instructions of run the instrument for as long as you can, just calibrate it on Monday, and just change the mobile phase, change the sample out, and then I want you to extract the mass accuracies. So this is the mass accuracy. How accurate is the mass that we are getting for this compound, leucine? It's an amino acid. How accurate is it in both negative mode? And then she switched the work list halfway through into positive mode, and she found less than 1 ppm error. This is pretty typical of this system because of that great electronics, great flight tube. And this is the flight tube for those of you who haven't seen a QTOF before. The ions travel this path before they hit the detector. It's how you get that high resolution data. And this is what I mean by collecting good data should be easy. You calibrate it on Monday, you keep running it, you should expect good data. But that good data also has to help you understand what the molecule is. And that's where measuring the isotopes comes into play. So our software puts these little theoretical boxes around the monoisotopic mass of pyruvic acid, which is negative ion mode. It's really, really small. So in the world of mass spectrometry, it's just a hard molecule to measure. But our system measures it really nicely, good resolution, good mass accuracy. And you can see the isotopic pattern fits really nicely to give you a confident identification of the chemical formula. That's key here. We want to know what the chemical formula is from the MS1 data. And you can see here, we have enough resolution that this third isotope, we're getting some interference. Like I said, there's a ton of signal in QTOF data. Some of it's not useful. Some of it interferes with your good signal here, but we have enough resolution on the system to separate this, which is excellent. And finally, it's the dynamic range. Being able to measure the small analyte, in this case serine, which is co-eluting with glutamine on top of it. Glutamine is a huge signal. I actually artificially spiked it in order to make this spectra work for my presentation. And you can see glutamine, huge, huge signal. Sometimes you can't manage the large signals in your sample that you're going to see when you're running real biological samples. Even when you have something very, very large like glutamine is here, not only should you expect off this instrument good mass accuracy of the large compound, you have good identification of something that's very small that is eluting at the same time, so it's in the same spectra underneath it. So this is serine, it's the endogenous amount, it's very, very small signal compared to the very, very, very large signal. This is the power of a QTOF. It measures the small stuff, even when the large stuff is co-eluting. So all of these features are why this is a great instrument for untargeted research. It also has flexibility, and this is more for reference. I'll send these slides out. It has flexibility of how you use the instrument. You can collect just the MS data. You can collect targeted, hey, I just want to go measure these targets. Please collect me very good MS data so I can get my publication out. But most commonly, we are also running an auto MS method. This is where we are asking the instrument to isolate precursors that are in a chromatogram and isolate them and then just send that precursor to be fragmented so we get that MS data. But do this independent of whatever, because it's an unknown. We don't want to program it to go look for stuff. We want it to do it for all the signals. So how we do that is a program called iterative MS. If you are building a metabolite library or a lipid library, this is basically where you're going to start. And we have automation that makes this easier. It's basically a systematic injection of the same exact sample, like a pooled sample that you've prepped from your study. And by doing it systematically, you basically are going to have the most MS rich data that you can imagine from your sample for your library building and identification. Why is that? Because the instrument still is limited on speed. So it can't isolate in one injection every single precursor, send it for fragmentation and MS data collection. So we say, do the same injection multiple times. Injection 1, you find the highest precursors, isolate those, send them off. In injection 2, we know at this retention time, we collected data on these 3 highest precursors, add them to an exclusion list. Don't go look for them the second injection. Don't send those off for MS identification or MS data collection. Send the next 3 highest. And it does this systematically, making a larger and larger exclusion list for data it collects until basically you have MS data for all the relevant signals in your sample. This is the fundamental of any metabolomics or lipidomics experiment when it's a new sample. But like I said, QTOFs are really nice because you have a lot of flexibility of the types of experiments you can do, as I showed you here, along with the pieces of software. But what I've done on this slide is basically outline kind of the top 4 things that customers ask from their QTOFs. I outlined kind of a basic question that you might want to know about your data. And I outlined as well my favorite acquisition and analysis techniques. So the most common one, as I mentioned at the top of my talk, untargeted profiling. I don't know what's changing in my sample set. I want to discover everything in it. That's where you'll basically do the iterative experiment, that library building on a pooled sample, maybe 2 pooled samples, like a treated and an untreated. And then you will collect MS, which is the most sensitive on every single sample that you have. You will use Explorer 2.0, which now has the MS identification and the statistical analysis component to it. Some people, though, routinely are measuring the same exact sample. They just want to know what's changing in it based off of some already known pathways that they have explored maybe in this workflow. First of all, this is a great experiment that you can move over to the triple quad that you have in your lab. But if you only have access to a QTOF, you can basically collect all the data the same way here, but you would use a different piece of software called the screener tool. I'm not going to go into that today just for the sake of time, but understand it's a great approach in making your data analysis life easier. Systems biologists, you want to know everything that's in a sample or matrix. You're going to just do that iterative example or iterative experiment, run the data in either Explorer or a Qual software, and then flux experimentalists. You're doing tracer studies. You're doping it with a high, heavy labeled compound in order to see how that compound is absorbed and metabolized in a biological system. You'll run MS on all your data and then use a program called VistaFlux, which is going to extract all the isotopes that are possible in your certain pathway and extract all that data and then plot it on a pathway for you to see what's changing. It's a great piece of software. All of these, I basically have a workflow slide for, but because the untargeted profiling is so popular, I'm just going to focus on that today. What does that look like in more detail? So let's pretend like you have no support from Agilent in terms of databases, but guess what? You do. You would basically come up to your QTOF, you have your chromatographic method, you have your pooled sample and your individual plate of samples. And the first thing you would do is take your chromatographic method and run that iterative MSMS experiment to get all of that really good MSMS data to build your library on, and you'd run that on a pooled sample or samples. If you're doing lipids, you would be using lipid annotator, which I will show you. If you're doing metabolites, you can use Explorer or Qual to basically build a library. You can have Metlin library, which Agilent has as an input, but basically by the end of this, you are going to have a custom library with or without retention times based off of just the MSMS data and your library input, like the Metlin library, which Agilent gives out. None of this has been, and I want to emphasize this, run against a standard unless you have that standard in your lab and you run that on your LC-MS method too. So all of these in terms of publication are still called tier 2 identifications until you run them against a standard. Still very useful for research purposes is collecting that high-res data, has all that isotopic fidelity and isotopic measurements, and you have MSMS. It's a great tentative identification until you can get a standard in. Your plate of samples, you would be running MS1 data on, which is the most sensitive, and running it through our Mass Hunter Explorer program. And at the end, this is where it's going to pull out all the features, and that could be up to 10,000 features. A feature is just a signal from the detector. And in the world of LC-MS, you have a lot of signal that is, some of it's biologically relevant, but only about 5%. According to some of the best researchers in this space, they did some meta-analysis of all the signal, and about 5% of the signal that you see is going to be biologically relevant. So this is going to extract all of that, do your statistics of your treated, untreated, et cetera, and show you, hey, out of all this signal, this is what's relevant in your 2 samples that are changing. Then you want to go and you want to identify just what's changing, let's say. And this is where you can do a lot of different plots. I'll show you those in a second. But you then apply in the same software your libraries. In this case, though, Agilent, if you are running an Agilent method, our Hillock method, our lipid method, we have retention time based MSMS libraries to support your research. So that's where you can apply, and all of those have had standards run against them. So you can apply as a first tier the Agilent Hillock metabolomics library, see what's identified, and then as a second tier, apply your custom library for your sample. This is the broadest way to kind of do a 1-2 punch of what are the metabolites in my sample, apply Agilent's library, apply your custom library, see what is identified. For those of you who are coming from a strict genomics background, you don't have this problem, but there will always, in the world of metabolomics with LC-MS, be stuff that's unidentified because of the chemical diversity that we're dealing with. At that point, you can take your unidentified features, and this is new in Explorer 2.0, which we just released in June. You can take your unidentified features, your MSMS spectra, and send it off to a third-party program called Sirius. This is a collaborator of ours for years and years and years, and it's the best software to tell you from the MSMS data what could that structure be. So a combination of these softwares is really how you attack a full-on targeted analysis. And you can see the complexity that can get involved. So this is where we do provide a lot more training on our end. But understand, this is the most complex it's going to be. It gets simpler from here. Once you know your sample really well, you characterize your sample really well with this workflow, you can move to a targeted method on the QTOF or a targeted method on the triple quad and do more of a routine analysis. Anyone doing lipidomics in the room should know about Lipid Annotator. This is a free software with our QTOF. It's going to take your MSMS data. Again, it's that iterative program I keep talking about. And it's going to basically do a library blast search of all your signals, looking at the molecular ion, the mass accuracy of it, as well as your fragmentation patterns, and apply an identification to it. Again, you haven't run a standard, so you don't know all the information about it yet, but it is a really good tentative tier 2 ID using this program almost instantaneously. This program is so easy to use. What I think is really important about this program that other programs like, this is a big differentiator between us and MS Dial, this will collapse all of your double bonds into one identification. So it's just going to say, for example, down here it says PG 36.2. It doesn't give any idea of where that double bond is. And so you're not going to over annotate if you're more of a novice to lipidomics. You won't accidentally over annotate your information just because you can't get the double bond information from an MSMS experiment unless you're running it against a standard. So I like that this program does that. I think it's valuable. But for that reason, when you run the same data set in Lipid Annotator, and then you might run it in another program like MS Style, which is free, it's great. This is free as well. You're going to have different lipid results in terms of how many lipids because they annotate the library differently. So keep that in mind if you start doing lipidomics with high-res systems. We have a program called Explorer 2.0 that is meant to do all your statistics and identification and feature extraction statistics and identification. And this is basically the big data analysis program that you'll use when you're doing high-res data. It's basically any omics researcher is going to want this. And historically, these programs are either hard to use or they take forever. And you can see our team did a great job at making the UI here much more workflow-based, much easier for a new novice user to start getting started with this. I still think that everyone doing this type of experiment should have some stats background, some knowledge of statistical analysis so that they don't overfit their data. But on the speed front, I'm very happy to report that Revident data in this program, because Revident data files are smaller, this program is faster. It's very, very fast. So you guys have a nice processing PC. This was an experiment I did for a conference. I was up against a deadline. I didn't have access to the PC that I wanted to use because a colleague was using it. And so I just went on a whim and I said, look, I'm going to run it on my laptop. It's the same laptop I'm presenting to you with today. It is an engineering laptop. It has 64 gigabytes of RAM on it. So I do want to be honest, it's not a small, dinky laptop. But I was very happy to see, I set this up on a Friday, went to go make my kid dinner, thinking it was going to run over the weekend, came back to check some emails, and about an hour later, it had finished. And this is unheard of on older iterations of this type of software from Agilent or free versions of it. It found in 50 data files, 10,000 features in under an hour. I was blown away. That type of a performance is really exceptional. So it can handle large projects. It can do that processing fast. And why are you doing this? Because you're trying to understand what's changing in a sample. You're trying to publish what's changing in the sample. You're trying to identify what's changing in a sample. When it comes to the publishing and the plotting, this can do a lot of different plots. This program can, and they're all high-res images that you can extract and immediately embed in your report or your paper or wherever you're posted for a conference. So you're not going to need extra programming skills or plotting skills to make a fancy diagram. You can get all of your plots from this program. Okay, that is wrapping up my QTOF portion because that's the biggest story today. It's the most complicated. It's going to get simpler once I move to the triple quad. But understand that everything you just discovered on the QTOF or any of the routine analysis that you might want to run on the QTOF, you can move those experiments to the triple quad. And the triple quad is really, really fast instrument that you can really push the limits of how many analytes you're investigating in one single injection. So that's the key with this triple quad that you have in your lab. And so keep in mind that metabolite, lipid, and peptide experiment I run on my triple quad day in, day out, and I switch between them. I do some flushing of the LC. So there is some like practical maintenance that I keep in mind as I switch from a metabolite project to a lipid project to a peptide project, but it's all manageable on the same LCMS system. Agilent has a simpler triple quad portfolio. It has 3 main triple quads. This is the Altivo. I bring it up because if you ever are in a space constraint lab and you don't have limits and you're looking at small molecules, anything below a mass to charge of 1500, this is a great system. It actually sits underneath that LC stack. A lot of people misidentify this instrument as a single quad. No, it's a really powerful triple quad. But the instrument that you guys have in your lab is a beefed up version of it. It is a larger footprint. It's a 6475. It can go to a higher mass range. So that's how we can do peptides on this system. And then the one above it, this is the 6495. This is going to have an ion funnel. It's also fit for purpose for omics studies. It just gives more sensitivity. But this is a very robust system, great for core labs trying to do all this omics research. So one of the things I just really want to emphasize here, it's the speed and reproducibility of what you can measure on a triple quad. Modern triple quads can go very, very fast, very reproducible, very sensitive. And so that's a benefit of using a triple quad. All of our triple quads also, you can remove the capillary or the MS, the ion guide, where all of your ions are flying into the mass spec. We have a really robust source, so this is maintenance that doesn't have to be done that often, but by being able to remove it and it doesn't vent the system, you can do an extra level of maintenance, get an extra level of sensitivity. Really great feature for core labs that have a lot of different samples running on them, where, you know, in a perfect world, everyone does perfect sample cleanup, but should something dirty happen to your, like get injected on your instrument, it allows a technician level user to be able to maintain the system and keep it cleaner. Cleaner means more sensitive. So this is what I mean by when I say we can go fast. This is a positive negative method. So I'm switching between positive ions and negative ions. I'm showing mouse plasma. I wanted to show something complex. And I'm looking at aconitic acid because it's 10 to the 4 signal, not too low, not too high. But you can see in the world of mass spec, we think about how many milliseconds do I sit on a signal before I start moving on to the next signal. And I mean, we are going at 0.5 milliseconds is the limit that my software team put on the system. And I'm going to tell you, when I did this experiment, I did it at 5 milliseconds as well as 50 milliseconds to really show where the limit is actually happening. And for aconitic acid, I'm seeing 1.7% RSDs for N equals 6 of a method that's switching between different polarities. This is unheard of speed for the system. That same reproducibility I'm seeing at higher dwell times or higher when you're sitting on each ion transition, each signal longer. This is how I know I can put hundreds of signals, hundreds of ion transitions or MRMs into one single injection and get good data. So let me show you one quick anecdote. We did a project with Phil Lorenzi. He was doing a drug study in mice, and he wanted to basically show, hey, this is a treated mouse, this is an untreated mouse. Show me the tox, in this case, it was a tox study. What are the pathways that are causing the toxicity to these mice? And so we ran for him the metabolite fraction, the lipid fraction, and the peptide fraction, and ran it with as many signals that we could fit and detect on our system. So that was 314 metabolites. Some were in positive mode, some were in negative mode. That dwell time was 2.5 milliseconds. I bring this up because I could indeed fit more signals in here. If I discovered something new on my QTOF in those samples and I wanted to bring that information into my triple quad, I can still fit more into my triple quad method. I'm not limited down to the 0.5 milliseconds yet. We ran a QC throughout his study and basically showed that 83% of those metabolites that were in the method had less than 20% RSD in that QC, so throughout the study. So it's actually a little bit higher than I was expecting. I was expecting something in the 90s in terms of reproducibility. But when I actually went to go look at, okay, these are the metabolites that are causing more, that are having more error, why? These are actually metabolites that are converting into one another over like the 24, 36 hours I was running this study for him, some of the metabolites were converting from one analyte to another. So you saw a linear increase in one, and its related one, you had a linear decrease. And this is basically showing, yes, the mass spec is working well, but I haven't fixed everything in metabolomics. I still could work on my metabolite stability on the sample prep side. But it's at least good because I'm running a QC, I know this is an issue, so I can just remove these from my statistics. Going to a much more stable compound, lipids, 99% were under 20% RSD, and that had a very fast method, 1.7 milliseconds for each transition. And peptides. Peptides, like I said, I had over 2,000 transitions in my peptide method. And this is a plot of that dwell time over time. So you can see the crowded regions of a chromatogram where you're looking to measure more and more are actually going to have a lower dwell time. And so you can see this one peptide having the minimum of the instrument, 0.5 milliseconds, and I can still see it very well with good points across the peak, so I get good integration. The peptides, high 90s, less than 20% RSD, so this is great. This is what we wanted to see. When you're actually looking at the results over time in this study, and I'm not going to go into the details, but we were able to see for metabolomics certain analytes that are meeting expectations of where the toxicity is coming from, but it was the lipids. It's combining the metabolites and lipids in one study that really showed, okay, where's the how can we potentially target the toxicity in a future study in order to reduce the toxicity? So this is me trying to explain that, or trying to emphasize that measuring more analyte classes, which you can do on the system, you can measure metabolites, lipids, peptides, all in the same sample set, will give you more understanding of your biology. So I encourage you, even if you're thinking, oh, I just need metabolites, maybe talk about looking at peptides and lipids, or vice versa. And then this is, because you guys have both instruments, it's really advantageous because you can go and discover something on the QTOF and then carry it over, optimize it on the triple quad with our Mass Hunter Optimizer software and measure it routinely on the triple quad. You get more sensitivity with the triple quad. It's just a different type of instrument. It's what it brings to the table. But really it's, okay, well, now that I know this signal in the QTOF, how do I get it to the triple quad? You can take a lot of the signal information and get a good enough result on the triple quad without doing any extra work, honestly. But if you wanted to get the perfect signal, we have what we call Mass Hunter Optimizer. This optimizer software sits in our Mass Hunter acquisition program where you're acquiring your data. It's amazing for taking a signal from a QTOF and optimizing it on your triple quad for the optimal voltages for measuring it. It's great at, if you bring Ben a method from a paper and you're like, hey, will you measure these transitions for me? And he doesn't have a method already for it, he can build a method faster using this program. It's where you can take a standard and optimize how to measure it for its precursor, its fragment, all the collision energies for it, what the retention time is on your chromatography. Although it does all of this automatically, it's still giving you reports that I'm showing here that have you double check the data before you go and run your full experiment. So it's giving still human intervention to say, yes, this is good signal, move this on to the final method. So this is just making the method building, how to program your triple quad faster, whether you're taking that from a QTOF, another high-res system that you measure your peptides on, let's say, or a paper, or a colleague who has another vendor system, or you're building a method from scratch, something that you just have a standard that you bought from Cerillion or VWR or wherever. Triple quad. The reason I'm spending so much time on the methods here is because triple quad data analysis, super easy. Everything is annotated already. So you just input your data into a program like Mass Hunter Quant, which is this blue icon on your desktop, or Skyline, which does small molecules, but it's really well known for peptide workflows. If you just input your data here, you're just going to integrate and check the integration of all your signals and then export it. You can do quantitation in these programs as well. I do a lot of programming with my profiling with my triple quad. So I just check integration here, and then I move it onto a statistical program like Metaboanalyst. Lastly, because I know peptides are such interest right now in the group, I wanted to emphasize that we have this really nice Skyline program. So this is using a triple quad to actually enable some level of discovery of your peptides that are changing in your sample. And we're really leveraging that speed of the instrument again. So basically, you tell your program, your Skyline program, what your peptides, what your proteins of interest are. It does a blast. It'll tell you what the peptides. Skyline is great at giving you from peptides to transitions to measure to injectable methods. And it does that all through the Skyline program. It's completely enabled with our Mass Hunter acquisition software so that you can have, it'll be several injections at first because the scope is thousands and thousands and thousands of peptides that you're going to want to screen for. And you just screen it on that pooled sample and you get nice sensitivity because you're measuring this on a triple quad. So I had one colleague do this last ASMS and I wanted to share it. It was 581 proteins that ended up being 1,700 peptides, 7,000 transitions. She ran this in a total, this screening method, this is the screening portion, in 14 injections that she ran on the same pooled sample. After the data analysis section of this workflow, she ended up with one single method that was 846 peptides, 390 proteins, with a total of 3,600 transitions in one injection. And that's the final method that she basically said, I can go screen all these proteins now in my sample set because I know from running this pooled discovery workflow using Skyline software that I can measure these peptides without running a standard against initially. So this is all, again, a tier 2 identification until you run it against a standard, but it's a really nice approach if you want to measure scope of peptides initially on a triple quad. It's really nice. On this triple quad system, you're going to have options more than just the metabolite and lipid method and peptide method that I showed you today. We're coming out we're coming out constantly with new methods on our exact LC and MS. So you can be doing quantitation of the TCA cycle. You can be measuring bile acids. You can be measuring organic acids with a non-Hillick method if Hillick is just not your cup of tea to start out with. These are all basically canned methods that Agilent can support you with. So really, if you have a sample set, you're looking for a specific compound class, looking to the triple quad might be the best first option in terms of the scope of the project, and Agilent has a ton of methods to support it. With that, I did touch on a little bit of everything today, really emphasizing our methods that are supporting our LCMS in order to help you get more biological information out of your sample faster. A lot of that, again, it's enabled with our consultants, basically scientists like me that help out customers like you guys, but reach out to me, reach out to Ben, reach out to and your Agilent representative, Dami, who's there today, because we just want to know what questions you have, how we can support you more. I did want to end on this. There are a lot of plant and food researchers using Agilent LCMS systems. I outlined my three favorite here. Mark Lang, he's at Washington State. Angela and I go way back. She's a big proponent of or user of our LC QTOF. And then Chris Elliott, he's food safety at University of Belfast, big name in food safety and authenticity. If you need to know some basics of how to approach your food questions, their ResearchGate is probably a great way to start, but you can also reach out to the consultants with Agilent. And with that, I will take any questions.
Perfect. Thank you so much, Karen. As promised, we do have a few minutes for questions.
I have a question. Thanks, Karen. It was a great presentation. I was wondering if someone wants to do a multi-omics method, does the order of the method matter?
So you mean like, yes, it does. When I approach multi-omics, so measuring more than one analyte, like say it's metabolites and lipids and peptides, like that one example on the triple quad I showed, we recommend starting with the metabolites, and then we flush the LC. We flush it with just some plain solvents, we remove the column, flush the LC with some solvents, and then we put our peptide column on. And then we do peptides next, and then we end on lipids. We find that is the best balance of the buffers on the system, as well as ensuring without doing additional maintenance that you're going to get the best sensitivity for the type of analyte. So if you're doing a multi-omics, approach it metabolites, peptides, lipids, but then you should be able to easily convert back, which I showed in my study, which has been published.
Perfect. Any additional questions either in the room or online? Well, please know if you do have any additional questions. As you heard Karen said, we do have a variety of representatives from Agilent in the room, so please feel free to approach them. They're largely at this table, so thank you very much to Agilent. But I do acknowledge that lunch has arrived, so I do want to share with you, we are moving into, it is time now where we wrap up the program for today. I want to thank all of our presenters again for their time, as well as Agilent for sponsoring today's event, and all of you for joining us today. Thank you to those in the room and online. At GIFS, we take pride in being a connector and a catalyst, and events like this are one way that we fulfill that role. Conversations like the ones we've had today are often where exciting ideas and innovative collaborations start. Of course, we want to keep the conversations going. So if you weren't able to get your questions answered, as I noted, or you want to reach out to us at the Global Institute for Food Security for more information, please reach out to us at info@gatebygifts.ca. And I do want to remind everyone, you have an amazing opportunity right now to tap into not only all of the technology that you've seen that Agilent has shared with us, but the amazing expertise that has been highlighted through Ben's presentation, and many of them are in the room today. So please take a moment, scan the QR code to view eligibility details, and submit your application. The recipient will receive a discount on strain engineering and DNA assembly services at GIFS. And as I mentioned earlier, for those in Saskatoon, you are welcome to join us for lunch, which is being set up at the side of the room. And for those joining us for a tour, you can meet us at the registration desk at your allotted time. If you don't have a tour time and you would like one, again, please visit the registration desk and we will try and get that arranged for you. And a representative of GIFS will take you over to our facility at your allotted tour time. A huge thank you to everyone. Thank you again to those online who took the time today. And please keep the conversations going. And thank you again to Karen and Ben. And we will allow people to break for lunch. So thank you very much.